Hi, my name is Kenita Diaz, and today I'm going to be talking to you about moth catching by spiders, specifically looking at the spreading behavior of glues produced by a subfamily of moth specialist species. Now, if you've ever played with a moth before, you might notice that they have a powder that flakes off on your hands. And so that powder is actually a layer of scales that coats their entire bodies. These scales provide the moth a number of evolutionary advantages, but the one we're talking about today is how they interact with spider webs. And so when a moth comes into contact with a spider web, the glue from that web attaches to these scales, and then as the moth thrashes, the scales detach from their body, freeing the moth from the web. If you were to look at this picture on the right, you could see this is a spider thread in the middle, and then it's coated with glue droplets. Those glue droplets, because of the super hydrophobic nature of the moth scale, don't spread very far and instead only attach to the moth and not actually the moth's base cuticle, its body. And so so this adhesive system is actually limited by the scales, which are loosely attached compared to the strength of the glue. And so there's a subfamily of species. One of them, one genus shown here, Pseudarachne, specifically the species Acarii, which have actually changed their webs as well as their glue in order to better catch moths. You can see a picture of them on the left. I think they're really cute. They kind of look like crabs. But on the right, you can see their web structure. And so this looks like a normal orb web, except it's been taken from its vertical position and made horizontal. That also allows the strands to be long and dangly instead of tight like they are on a normal web. But if you look at this picture, you can also see that there's white that's all over these strands, and that's actually the glue that you're seeing that these spiders present. One of the things we realized in our previous research, however, is that the glue is not innately amazing, at least not off of the moth scales. When you bring it into contact with a glass substrate or something more traditionally used for testing, you'll see what we call simple linear flow, where the glue droplet comes into contact with the surface, and then the glue spreads radially outward from the center, um, in this shape. When we bring the glue droplets into contact, however, with a moth surface, like one shown here, we'll see a droplet at the top right and one down here. You can see what we call the complex nonlinear flow. If you look along this front, you can see it's no longer straight, but instead it's going in waves and seems to be going down some type of patterns. If you look right here, you can actually see the scale that's left over after the front, and it's actually filled in at the end. And so some added forces are actually pulling the glue in different directions. It's no longer spreading straight linearly outward. So if we were to look at that scale more closely, we'd realize that there's actually a microstructure on there. It's not like a flat surface. It instead has all of these long canals that run along it. And this is something that we think is important for the flow of the glue. Not only do we have individual scales which are controlling some of this spreading, but we actually have scales that are all together, right? There's a colony of scales that are all being produced. And so, for instance, in the wing, you can see all of these scales going top to bottom. They're at varying angles. They're all actually of varying shapes and sizes. And so one thing that I noticed while doing this, while working on this project, was that the base cuticle right here, which has had all the scales taken off, is actually very dynamic. And so scales actually grow out of sockets. And those sockets are arranged in rows, but the rows aren't very regular. They alternate up and down. To say they're rows means they kind of just run the length of the body. What I did realize, though, that's very, very important is if you look at how close the sockets are to one another, they're clustered and they're actually on top of each other. They're not like shingles on the roof that are laying directly next to one another. They're overlapping in a multitude of ways. If you were to look at, say, the size of the scales that are present on the screen right now, one thing you might notice is that if they were all to be growing out of these sockets, they would overlap one another in a multitude of ways. So there's a lot of interaction between these scales. And so what we want to start doing was actually describe the spreading behavior. So as we did before, I'm going to show you a video of the spreading on the surfaces. So we took some high-speed videos at 1,000 frames per second of pseudarachnoid glue droplets coming into contact with the surface. We then used Canovia to track the paths of those fronts, and we used 4 for each droplet: north, south, east, and west. And you can see how they're moving right now. What you'll notice is, as we track it, we have that simple linear flow. Now, when we look at the spreading behavior of pseudarachnoid glue droplets, you can see that this behavior is no longer linear. In fact, there's a multitude of different fronts that we can track if we're going on any particular side because of how dynamic and strange it varies from point to point along this. One thing that's also important if I let go of the video is you might see that this video lasts significantly longer than on glass. Because not only is this behavior dynamic on the Moth scales, but it's actually much, much faster. And so if we look at the spreading behavior, we have graphs like this. So we have our 2 glue droplets from this video, and then we have our 4 points we're tracking for each one. And what I've done here is I've actually created a gradient. And so the beginning of spreading occurs at red, which is 0 seconds, and then goes all the way to the end of spreading for our video, which is half a second. Now, the only point in which this behavior is not linear is when the droplets come in contact with one another and flow sideways, as we've seen before. You'll see that in this video happening right here. The glue doesn't go over each other, but instead turns and changes direction. So besides that, it's very, very linear. If we were to look at the tracking on the Moth scales, however, we see what we already know, right? This behavior of the glue going at very wide angles, constantly changing shape and flowing. Another really important thing to note about this is that if you look at the scale bar, we have this gradient once again. Red is once again at 0, but the blue only goes up to 0.04 seconds. And so this is spreading 10 times as fast as it was in the other videos. And so how do we make a model of the glue behavior on these scales? Well, first we need to look at the microstructure. As I said, there's these different types of canals that are running along the body of the length of the Moth scale. And so one thing we can do is create a spatially explicit model in which we have squares that distinguish different regions in which we think different equations or aspects of the glue may dominate, changing the flow pattern. But one thing that's interesting to know is if you look down these canals of the length of the Moth scale, you'll see that there's pores in them. So there's actually a microstructure within them at even the smallest scale. Now, if we look at this allows us to define the scale thickness as well as the dimensions and the ridge depth, which will be important for documenting scales. But what we also have is we have a macrostructure. We need to describe how these scales are sitting relative to one another because that will change how the glue is flowing. And again, we can break up this larger structure into its own set of scales, a set of boxes, looking at how these scales overlap. Looking at this structure, one thing we can do is it allows us to define the scale density and the relative angle and orientation of the scales relative to one another. And when we combine these, we actually create a 2-layer system because we're going to have behavior of glue flowing underneath the scales, this bottom base layer, and then on top. And so by combining them, we can look at a more dynamic effect. Also, it's important to note that these two are not mutually exclusive. Glue that's on top will flow to the bottom, and so what is on top will actually affect what is happening on the base layer. And so when we look at this, this has us break up our equations to what we believe will be 2 spreading phases. First, we have that surface flow, which is the flow directly on the top of those scales. And if we look right here on the right, if you look at these depths of the canals, you can actually see that there are these speed bump type of things in them. So not only would glue be flowing down them, but there'd be some inhibitory force there. So we talk about this as it seems most likely to talk about pipe flow. It seems that they're going down a kind of canal or half pipe. And so this flattens the scales, which will affect our second phase. But one thing that's very important is we think that because of the way that the physics of this works, the interaction between the glue and the surface is actually a resistive force. Primarily, the flow is going to be dictated by the viscosity of the glue and its innate inertia. However, this behavior completely changes once we get to our second phase, which is what we call matrix flow, or the fact that these scales exist as a meshwork overlapping one another. So as the glue flows over them, we have the pressing down of scales, which actually lowers the amount of space there is to fill. Think of this as when you are using a garden hose and you put your finger over the end, you're actually creating the same amount of water trying to go through a smaller space, which makes it go fast, which makes it go much faster out. And so this is increasing in our case, what we call capillary pressure, or the fact that the surface tension of the glue, in this case, is going to be more attracted to the scales than itself, which actually creates a passive pushing effect. And it increases both pipe flow speed, things like that. And so as capillary forces begin to kick in between these 2 droplets, you have an increase in spreading, and all of a sudden this surface tension that was inhibiting the glue droplet spread on top, trying to pull it into a ball, is actually become a propulsive force, which will change the spreading, so leading to leading to an acceleration at some point. And so in this case, in the second phase, contact between the glue and the surface actually becomes a force that elicits flow. And so you're going from an inhibitory to an eliciting flow. And then another thing that's important to know is that when the scale, if you have this half pipe, say, structure on the scale, when you bring two of them into contact, you no longer have a single hole. You're more creating something akin to a full pipe. Obviously, it's not perfect because they're slightly off, but this would allow more glue to flow in general. And so when we're looking at this, we're trying to model the behavior. We have a few different equations that we could use. We have the Hagen-Poiseuille looking at pipe flow, again, in that first phase. And then we have this capillary flow and capillary action that's occurring underneath the scales in the second phase due to pressure. One thing we tried to do is look at how some of these models should estimate the spread of our surfaces. And so we compare it to something as simple as Tanner's law, which is just spreading on a flat surface. And so what we see is that when we look at the volume flow rate estimated by Hagen-Poiseuille or capillary pressure, they both fail to show the very large and dynamic spreading of pseudoarachnoid glue. And so one of the things we're thinking about is not only do these models have to be spatially explicit, but it will probably need to be a combination of these models to properly show the force that we need. And so I'd just like to acknowledge our collaborators at the Museum of Natural History, Cheryl Hayashi and Richard Baker, as well as our collaborators from the University of Tokyo, Tanikawa, who gets us our spiders and has some of those pretty pictures I showed you, and then the FAU Biomechanics Lab and Imaging Lab for helping us take pictures of these moth scales and do these SEM estimates. So we'd like to thank Dr. Marianne Porter, as well as Braden Rudy and Aubrey Clark, as well as the National Science Foundation for funding.